We have a number of things that we still need to consider with Mendel. As we've mentioned, he had some serious problems with the disappearing and reappearing of traits in his generations. We looked at the crossing of the F1 generation. So in this instance, starting with a yellow round and a yellow round. And we saw that that actually gave us something that you would not normally predict. If you're crossing two individuals that are yellow and round, you would normally expect yellow, round offspring. Well, we know that that's not true. So he started to see traits that were not present in the parents, right? In particular, he was trying to figure out how you can go from two yellow round pea plants to getting a plant that produces not only green but also wrinkled. So wrinkled falls into a second trait category for us, something that we refer to as a dihybrid, so two traits, when we're considering the genetics behind it. So back to our chromosome picture from before, this of course is just focusing on the dominant and recessive of our yellow and green characteristics. So remember, dominant is yellow, big Y, recessive is green, little y. But what does that mean if we start considering more than one trait to help Mendel explain this disappearing and reappearing of traits? We have to consider something known as a dihybrid cross. So this is looking at two traits instead of just one. And so what Mendel had to do was actually make sure all of his traits that he was interested in were included in the parental generation. So he started with two plants, one with round yellow seeds, one with wrinkled green seeds. So these are all the characteristics possible based on the traits he was looking at, color, and we'll call it seed texture. So whether or not they were yellow or green, and whether or not they were smooth or round. Round is the same as smooth. Or wrinkled seeds. In his first generation, he had the same problems he's had all along. He gets essentially one set of traits. He got all plants with round yellow seeds. So where did the green go? And where did the wrinkle go? Well, as you might predict, it comes back in the next generation. When he crosses two of the F1s, he gets all of his characteristics again, round and yellow, round and green, wrinkled and yellow, wrinkled and green. So if we look at it as a dihybrid cross, remember when we had our monohybrid cross, we had a nice small 4-square box. Well, now when you deal with two traits, you actually develop a 16-square box to look at all of the possible combinations of gametes and the next generation. I'm not going to get into all of the details of building those dihybrid crosses with you today. If you're interested in that, we can talk about that more later on. But the biggest thing we want to focus on is the phenotypic ratio that is associated with this. So the phenotype that you get with every single dihybrid heterozygous cross is a 9:3:1 ratio for the phenotype. Now, if you want, you can go through this table on the right and think about all the possible genotypes that would go along with this. We're going to focus on the phenotypes. That's what Mendel focused on. Remember, he had no knowledge of genetics per se. So we'll focus on the phenotypes. What did they look like? So what did we say a heterozygous dihybrid cross would produce for the phenotypic ratio? So this is going to include yellow, green, smooth, and wrinkled seeds. When you do a dihybrid cross, you end up with 16 blocks. So keep in mind, that means that you need to have 16 individuals that you consider in your ratio. What did we say that ratio was going to be? Right here. 9, 3, 3, 1 every time. Because he started out with an individual in the P generation, we'll say P mom, was dominant for both traits. But we don't know if mom was a heterozygote or a homozygous dominant. P dad is recessive for both traits. How do I know that? Wrinkled is recessive, green is recessive. So crossing those two, this is actually set up something called a Testcross. If you have the expression of a recessive trait in an individual, they must be homozygous recessive for that trait. We'll look at this. Mendel used this to develop several sets of laws. The first one is the law of independent assortment. We've already talked a little bit about the law of segregation. This, the law of independent assortment, actually considers something that Mendel didn't have any idea about. It refers to metaphase 1 of meiosis. This is the separation of the chromosomes during meiosis 1. Mendel didn't have any idea about these genetics, remember, but he knew that the pairs, not the genes, but the pairs had to assort independently, meaning that when you form gametes, the chromosomes that are placed into those gametes are random. And it means that if you carry big Y, little y, those are going to split up, and one goes to one gamete and one goes to another gamete. We mentioned this before, just kind of reminding you where that terminology comes from. So how did he start looking at this even more? Well, we look at it today a lot in dogs because dog breeding has become a bit of an art with individuals. This certainly falls under something we'll call later artificial selection. We are selecting particular traits in the dogs that are of interest to the breeders. But he set this up so that he could consider what the genotypes would be for these individuals, considering their traits. So one dog, this last one over here, is chocolate coated and blind. These are both recessive traits, as you see by the indication of the little letters, right? So what he didn't know was if this chocolate lab with normal vision was big N, or big N, little n, because remember, both of those genotypes will give you the same phenotype. This will both be normal vision, right? Black coat could be big B, or big B, little b, both of which will give you a black coat color in this case. Okay, so what he started to do was cross these dogs and think about how he could get a phenotypic ratio of 9:3:1 so that he would know what the original dog was, whether or not the original dogs in the cross were heterozygotes or homozygotes. Today we do this with genetic testing, but for many, many years it was trial and error. I mentioned the term Testcross to you. So this is mating an unknown individual, okay, with another individual that is homozygous recessive. This is the critical part. You have to have an individual that's homozygous recessive. Why? Because in our crosses, when we set them up, we have to see whether or not we're going to end up with offspring that show a recessive trait. If you crossed our black-coated dog here, remember we said that could be big B, or big B, little b. If you cross that with a recessive dog, little b in this case, if you set that up in our normal crosses, they've simplified this table for you, but you could have little b with big B like we've done before. And every block is big, little. So all of our dogs, as they're showing you here, come out to be all black, big B, little b. But if you cross this homozygote recessive with a heterozygote, as they've shown you, and if you would set this up the way we used to so you're comfortable with it, big B, little b, but then you get little b, little b. So you'd get a 1-to-1 ratio of dogs. So by backtracking from the offspring, you can tell a lot about the parents. And this is test crossing. We see this all through genetics. Like I said, today we do this more with testing, genetic testing, but you could certainly set up a test cross of any organisms as long as you know which traits are dominant and which are recessive, and that you start with an individual that is homozygous recessive. That's required for a test cross.